Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, with our ongoing series on introduction to proteins, today we would be discussing on protein degradation, and under protein degradation we would be specifically talking on ubiquitin proteasome pathway. And for this discussion we have once again with us in our studios Dr. Unnati Gulati. Dr. Unnati Gulati is academic coordinator with CEC Consortium for Educational Communication. So let's welcome Dr. Unnati Gulati and let's try to grab maximum knowledge through her. Dr. Gulati, welcome to the research lecture.
Thank you, Geetika. Welcome dear students. Continuing our series on proteins, we have discussed about the protein structure and importance of the 3D protein structure. We led to the misfolding of proteins and then we talked about the diseases that can be caused by misfolding of proteins. So once the protein has misfolded, is there any redemption for it? What are the mechanisms and processes that occur in a normal cell to regulate such kind of misfolded degraded proteins? So today we are going to talk about such mechanism. One is the ubiquitin proteasome pathway and the other is the lysosomal pathway. In this lecture we will be dealing with the ubiquitin proteasome pathway. So as we have discussed about various misfolding that occur in a protein and how that can lead to loss of function because there is loss of structure. By accumulation or aggregation of such misfolded proteins, it can lead to gain in toxicity. We have heard about the neurodegenerative diseases where plaques and fibrils may occur. We have also talked about accumulation of protein aggregates which can cause lot of burden onto the cell and also result in accumulation and chemical aging of the cell. So on the hindsight, we had looked at how Alzheimer's, Parkinson's disease all are having a characteristic misfolded protein which results in aggregates that cannot be folded back again. So when a protein undergoes this pathway of folding, there is a chance that among this pathway only it goes off pathway and may form aggregates or may get misfolded or may not form the proper complex or quaternary structure that is needed for proper functioning. So what happens to them? All of them are transferred to the protease pathway, either the ubiquitin proteasome pathway which will take care of 80 to 90% of such proteins, while the lysosomal pathway will take care of 10 to 20% of the cell organelles, extracellular proteins and some intracellular proteins as well. On a cautionary note, not all these fibrils and plaques can be reverted back to the normal state by these proteasome pathway, yet lot of proteins which have misfolded or have lost function or are not required by the cell at the present metabolic state can be taken care by these two pathways. So degradation may sound as a term, but this is very essential for regulating several cellular functions. Many proteins which are active during stress or difficult conditions in the cell can adapt to particular sets of conditions, and these proteins may not be required when the conditions are favorable again. So these proteins do not have any function under that state and hence they are also degraded. When the cell goes through the cell cycle, there is deactivation of inhibitors of several inhibitors which are functional in mitosis after the chromosomes are aligned to be separated to the opposite poles. So the sensitivity to proteolysis suddenly will increase when the inhibitors are going to be inactivated and then the cell will move towards the cell cycle. So there are several factors which are also required during DNA synthesis that need to be accumulated or removed at a very quick pace. Hence there also degradation of these factors and inhibitors is required. Many a times misfolded, mislocalized or excess proteins or damaged proteins can be taken care by degradation pathway. Turnover and recycling is a very important cellular process and it is in fact very much required for replenishing the amino acid pool which will be responsible for creating new and new proteins as and when metabolic needs arise. So there is going to be turnover. The E3 factors and the allosterically regulated transcription factors, several enzymatic reactions, they all require energy and this regulation which is ATP dependent, you must understand is required at all stages of development. And when we talk about energy requirement, we must understand that this would be a very specific and a very important process. So, recycling is, as in our lives, also important in a cell's life. So, there are several zymogens and prohormones that need to be activated, and there also degradation is important. We know about the cleavage of N-terminal methyl to form the methyl peptidase. In chymotrypsin, where chymotrypsin protease is going to create trypsin, several viruses which are occurring as precursor proteins can be cleaved, and these proteins can then become functional and can result in the infection. Also, when we are fine tuning the metabolic processes, many a times gene expression level, when we talk about transcriptional factors regulating the expression of any protein or how much amount of protein should be present, that type of process may be further fine regulated by degradation kind of a process. Come to think of it, when we talk about going all the way from DNA translation, DNA transcription to translation, and that process resulting in amounts of protein cannot be regulated as quickly as we can regulate by degrading the amount of protein that has been created. So, we are going to talk about protein turnover and stability. Proteins, as were assumed to be once very strong molecules which do not get degraded, were again this hypothesis was broken down when it was understood that proteins are also dynamic and they do undergo degradation and resynthesis. Proteins are always having a half-life, or they are not always going to survive when we are talking about fine tuning. So, you don't need all the proteins apart from the structural proteins which do also undergo some kind of recycling. So, you need to recycle the proteins at all stages. So, the proteins have a half-life, and it may range from 0.2 to 150 hours depending on the function the protein is a part of. Many proteins are fast turnover, means their concentration is increased or reduced very quickly, and there also degradation is important. Many times it has to be activated or inactivated. Transcriptional factors, enzymes which control the critical rate limiting steps in biosynthetic pathways, and several cell cycle proteins and inhibitors and regulators are fast turnover proteins of which show very low half-life. Then there are several structural components or structural motifs which are present in the protein itself that suggest that this protein is either a long-lived protein or it will be degraded quite faster. So, there are some key pointers. If there is an N-terminal amino acid residue, then we are going to call it the end rule, and the residues which show higher propensity towards degradation, if they are present. So, these are methionine, serine, alanine, threonine, valine and glycine. So, if they are present on the N-terminal, then this protein has chances of surviving more than 20 hours. If the protein has phenylalanine, leucine, aspartate, etc., then the protein will have a half-life or of 3 minutes or less. So, there is a wide variety of wide range of life span of a protein depending on its function, which also is guided by its structure and its primary sequence. Here are some examples of the lifespan of a protein. So, you have ornithine decarboxylase which is only 0.2 hours, while you have cytochrome C which is 150 hours, and then lens crystalline which will survive all through the life of a human being. So, there is a wide variety of lifespans depending on the function and the structure the protein has. Another sequences which are found in the protein mark it for degradation. These are called the degrons, and you will find that these PES sequences having domains which are very rich in proline, glutamine, serine and threonine will be rapidly degraded, while glycosylation of some PES sequences can prolong their lifespan. and there are certain degrons which I was talking about, or destruction boxes, which are present at the C terminal, and these are small 9 amino acid long sequences which mark the protein for ubiquitination and degradation, and this is seen in cyclin A, B, and you can see they are very much regulated. So, protein homeostasis is maintained in the cell all through. There are several machineries that are working towards it. There is a balance that has to be created between synthesis and functioning of the cell of the protein, and then its degradation. So, there is a protein quality control system which is in place. It will also encompass your chaperones which will mend the misfolded proteins. It will also encompass the 2 pathways that we are going to talk about, which will reclaim back the amino acid pool from the degraded protein. There are several protein quality control or management strategies. You can upregulate quality control components. The quality control component, first thing is the chaperones, which can be of both chaperone and chaperonin type, we have discussed before. So, these there can be several transcription factors which will manage stress, enhance expression of the molecular chaperones, and degradation will be reduced. There are several factors which cause degradation of the misfolded protein, and they will be enhanced when they have to reclaim back the degraded protein, or these degraded proteins can be sequestered back and put back into the pool of free amino acids. The key enzyme, the key protein here is the ubiquitin. It is called ubiquitin because this is one protein which is found in all organisms. In prokaryotes, you will not find a ubiquitin like molecule, but yes, there are precursors of a ubiquitin like structural ancestor which is prevalent in prokaryotes, while in all others, as you will see, this ubiquitin is conserved sequence, only varying at very few amino acid sites. I would like to say here is that yeast and humans have a very similar ubiquitin, varying only at 3 amino acid residues out of the 76 amino acid residues comprising the ubiquitin. So, we are relatively very similar to yeast in terms of the ubiquitin, and hence this mechanism being so conserved across the animal and plant kingdom suggests that this is a very selective and a very important process which has not evolved much. So, this is the ribbon diagram of the ubiquitin protein, which is fairly small protein, and the key residue, key site here is the C terminal, which is the free end. This is a 85 kilodalton protein, very small, 76 amino acids, and it has a very characteristic beta grasp motif. This beta grasp motif also you will find in the prokaryotic ancestors as we were discussing. This beta grasp is consisting of a 3.5 turn of an amphivathic alpha helix and a short 310 helix packed against a 5-stranded beta sheet, and the core of it has 16 to 17 hydrophobic residues, which gives it lot of capacity for hydrogen bonding. The C terminus is very interesting here. It has a glycine-glycine chain. This is free chain which is present to form by bonding with the other ubiquitin or target proteins. And this ubiquitin is synthesized as a precursor along with several ribosomal proteins, and ubiquitin occur as tandem units. It has to be further processed. Ubiquitin is a Nobel Prize winning molecule. It got the Nobel Prize to Aaron Scheisenover, Avram Hershko and Erwin Rose in year 2004, and this very small molecule has changed the course of how we are going to look at diseases, and down the lecture we will discuss how ubiquitin is important for several diseases. Come to think of it, metabolic, cancers, virus, and even your neurodegenerative and myopathies. So, ubiquitin is a Nobel Prize winning material, very small protein but very crucial. When we will look at it, it has its role in regulation, it has role in degradation, and lot of signaling is ubiquitin dependent. So, this quality control of nascent protein, membrane trafficking, cell signaling, cell cycle control, X chromosome inactivation and the maintenance of chromosome structure. So, almost all the processes do require ubiquitin signaling in some form or the other. A characteristic bond that we will encounter when we talk about ubiquitin is the isopeptide bond. Isopeptide bond is different from a normal peptide bond because this is going to be formed among amino acid side groups which are not present in the main chain in the backbone of the protein. So, amide bond from the carboxy terminus of one protein and the amino group of the eta lysine. So, lysine has several NH2 groups which will give rise to, which can participate in the isopeptide bond formation. So, generally the glycine free at the C terminal. So, one of the glycine participates, provides the carboxy terminus and it forms the isopeptide bond with the lysine of the condemned protein. We are calling the condemned protein because ubiquitin may be for signaling or for degradation. And these isopeptide bonds generally will be formed between lysine 48 eta amino acid of one ubiquitin and C terminal of the glycine 76 of the following ubiquitin when we are talking about a poly-ubiquitin chain. So, this is what it looks like, a ubiquitin carboxy terminus forming a isopeptide bond with the lysine eta group NH3 and giving rise to this isopeptide bond. Why are we going to encounter poly-ubiquitin? Several ubiquitin molecules linked together in tandem with this glycine and lysine isopeptide bond is called a poly-ubiquitin. This can be 1 to 10 to even 30 such ubiquitin molecules in tandem attached to the protein. These are very crucial. Lysine, not just 48, but lysine 6, 11, 29 and 63 of the ubiquitin also can participate in this kind of long chain formation. Very crucial is that for recognition of this ubiquitinated protein to the proteasome, as we are saying, this is a ubiquitin proteasome pathway. So, once the protein which has been tagged with this small protein ubiquitin has to be recognized by the proteasome pathway, and it is only recognized when there is a short chain of 4 ubiquitins bound one after the other. Anything smaller than this will not be recognized. Also, this kind of polyubiquitination provides a structural motif, provides lot of hydrophobicity which is very easily recognized by the 19S subunit of the proteasome. And there can be several ubiquitinations which can occur at lysine 6, 11 or 29. They will also target the protein for proteasome. And here you can see that these lysines 11, 6, 29 etc. of the ubiquitin give their eta amino group, and it can form with the following glycine of the second incoming ubiquitin, and the bond is formed, and this will give rise to a again isopeptide bond, but can give rise to a chain of ubiquitin, as you can see here in a ribbon diagram of ubiquitin. So, this is one ubiquitin and the following ubiquitin with its lysine of the first and glycine of the second. Here also C terminal is responsible for forming this bond. This polyubiquitination again give rise to a new kind of vocabulary when we talk about protein signal and recognition. And another vast area of research that has now opened up is the ubiquitin code or ubiquitination code. So, there are possibilities of several lysines which can form, which can bind other ubiquitin molecules, or ubiquitin can bind through other lysines to the target proteins. There can be multiples of ubiquitin, there can be branched chain of ubiquitin, there can be single chain of ubiquitin onto the substrate. So, you have mono ubiquitinylation, you have a mixed ubiquitin chain, you can have a homogeneous chain, or you can have a branch chain. all of these have now been found by various techniques that they have roles in signaling, they have roles in targeting. So, more and more we explore this code, we will understand that there is a new kind of code in protein targeting and signaling, apart from the genetic code that we know. So, there is a code which is again looked after by researchers and scientists with lot of curiosity. So, as we were talking about that there are several lysines which can contribute, and any lysine which is contributing can in fact change the target and the destiny of the ubiquitin molecule, so of the target protein. So, lysine at 48 will definitely channelize it to the proteasome, while a lysine 6 linkages will be found when mitochondria are undergoing damages. Lysine 27 linkages are found when DNA repair and autoimmunity is undertaken. Lysine 29 has been found in again proteasomal degradation, while lysine 33 is found for regulating the protein or protein trafficking through the trans-Golgi network. Even the methionine 1 of the ubiquitin is sometimes found to be responsible in the NFK beta activation, in immune response and regulation of interferon production. So, all these are key areas of research in which lot of work is going on. So, the two pathways that we are going to look at for protein degradation, one is the ubiquitin proteasome pathway. It will require ATP and hence it is a very sensitive pathway. The ubiquitin, which is a 76 amino acid, will tag the cytosolic proteins, the nucleoproteins, and it will target it to the 26S proteasome. Lot being said by the ubiquitin, it is actually not the degrading molecule but only the tagging molecule, and it can vary its signaling by an amount of or the type of branching that will occur when lot of more and more ubiquitin molecules are attached to the target protein. While we will also encounter the lysosomal proteolysis, in which we will talk about several enzymes which are going to be present for degrading the protein. There are going to be several chaperones which will bring in the proteins and either unfold them or degrade them. We will also talk about lysosomal proteases and how lysosomes have environment for degrading the proteins in the next series of lectures. So, today we focus on the ubiquitin proteasome pathway. We have discussed, so this brief diagram is showing you how these two are significantly different and what all components are present. So, today let us focus on the ubiquitin pathway, and here ubiquitin is only the tagging molecule and the actual degrading moiety or component is the proteasome. So, we will look at this barrel like structure and let us discuss in detail what does it mean. Ubiquitin itself is a tagging molecule, but it comes with lot of enzymes that are required for selectively tagging the proteins, and in this process there are the E1, E2s and E3s which are the activating enzyme, the E2 which is a conjugating enzyme, and E3s which are ubiquitin ligase. A note here would say, I would note here that E1s are only 1 and 2, while there are 500 to 800 ubiquitin ligases. These are very important for selectivity of the protein which has to be ubiquitinated. There are other enzymes also which are de-ubiquitinating enzymes, and they are also responsible for regulating and fine tuning the protein concentration. So, the ubiquitin system is constituting about 5% of the genome and it comprises of the accessory enzymes. We will also in this lecture talk about what is the role of these enzymes and how they can be manipulated for various drug discovery and therapies. So, briefly, the ubiquitinylation is a tagging of the proteins which are mapped for rapid degradation by the covalent attachment of several molecules or of ubiquitin. So, first there has to be creation of a pool of ubiquitin which has to be activated. and activation is carried out by the enzyme E1. E1 is the enzyme which hydrolyzes the ATP. It transfers the AMP group onto the C terminal of the ubiquitin, and this AMP with the ubiquitin becomes the activated molecule. It is a high energy bond, and this is further transferred to the E2 via moderation of a cysteine. So, the E1 enzyme transfers the thioester linked ubiquitin to the ubiquitin conjugating enzyme E2. Once the E1 has formed the activated ubiquitin along with the AMP, it transfers it to its own cysteine, and this thioester bond, once formed because of the high energy released from the removal of phosphate from the ATP, is now transferred further to the E2 through the cysteine again. So, the movement of one thioester bond to the second thioester bond is now without requiring any energy, because it is itself a high energy bond. So, this transfer is occurring. Now, the ubiquitin which was on E1 in activated form has moved to the E2, which is the conjugating enzyme. This is ATP independent reaction, and this E2 will now reserve the pool of all the activated ubiquitin. So, as there were only 1 or 2 E1 enzymes required to activate all the ubiquitin molecule, more and more as we go towards the selectivity, we will find more and more E2s and E3 are going to be part of this pathway. So, the E2 associated ubiquitin will now reserve the pool of activated ubiquitin for attaching it to the various target proteins for either degradation, signaling or regulation. So, once it has come onto the conjugating enzyme, the ubiquitin is going to be linked to another thioester bond to the active site of the cysteine in the E2. There are 4 different type of E2s, about 30 such molecules of E2 can be found, and they have a similarity that they have a core domain, and some of which have short C or N terminal extensions that are involved in E2 localization or in protein-protein interactions. These different E2 enzymes are able to interact with the overlapping sets of E3 ligase. As you can see, once the E2 has a activated ubiquitin here shown in the green, it will transfer it to the E3s. Here protein-protein interaction between the E2 and the E3 has to occur for the movement of ubiquitin from the cysteine of the E2 to the cysteine of the E3. So, multiple ubiquitins can now be added from the E2s to the E3, and a polyubiquitinated protein can be formed. Ubiquitin ligation is the third step involving the E3 ligase enzyme. This will transfer the ubiquitin from the E2 enzyme to a lysine on the substrate protein. Here again it is going to form a isopeptide bond between the substrate lysine and the C terminus glycine of the ubiquitin. So, substrate selection and specificity is the challenging role which we are now going to look at very curiously for various research and disease therapy purposes. So, these E3 ligases are responsible for substrate specificity and regulation of the ubiquitination process. About 100 E3 binding domains are found within the proteins that are going to be recognized by the set of 500 to 800 E3 ligases. So, E3 ligases are responsible for further addition of the ubiquitin by ubiquitin elongation enzyme E4. So, 7 lysine residues in ubiquitin are present, and they can be ubiquitinated independently or in a chain or a branch format, and this enzyme E4 is responsible for elongation and multiple ubiquitin formation. Introduction to proteins and protein degradation and ubiquitin proteasome pathway and scope of research. We have talked about ubiquitin being tagged onto the protein. There is an existing code of ubiquitin onto the protein that will target it for signaling, degradation or regulation. So, what are those signals which are present on the protein itself that will invite that it has to be targeted for ubiquitination or it has to go to the proteasome for degradation? So, there has been defined as degron or the destruction or degradation signals which are either primary sequence or structural motifs existing onto a protein. So, these are degradation signals, or you can say the red flags present onto a protein which will invite any kind of protein tagging. So, degron or part of a degron will be recognized when it is either bound directly by a specific E3 or E3 E2 complex, and it will then be further ubiquitin will be attached onto it. Here for example, we are seeing a phosphate dependent degron which is a 2-phosphate attached to the beta catenin unit. So, here phosphate catenin functions as, so the beta catenin functions as a key transcriptional regulator during Drosophila embryogenesis. So, during development it is constitutively phosphorylated, that is 2 phosphorus are attached to it. So, this protein will be constantly ubiquitinated and degraded until the phosphorylation is removed, hence the protein will not be recognized by the E2, E3 complex and hence will not be channelized for the degradation. So, this kind of signal or sequence found in a protein regulates its going towards a degradation pathway or remaining active and performing its function. Several SH2 domains and other coil kind of motifs can also act as substrate binding sites for E3, or they invite the binding of E3. So, E3 ligases will have the SH2 binding domain, and once they recognize, they will ubiquitinate such a protein containing the SH2 domain. And even exposed hydrophobic patches which are critically sequences in the which are critically structurally found in the misfolded aggregated proteins are red flagged and are recognized by the E3 and pushed towards the proteasome pathway. So, many a times multiple phosphorylation dependent degrons which are found in yeast inhibitors of onset of DNA replication. So, in this it is crucial that there is an all or none phenomena. Out of the 9 sites, 6 at least must be phosphorylated so that the cyclin dependent kinase can be either recognized by the E3 or not recognized by the E3. So, there has to be 6 sites must be phosphorylated for it to be degraded, otherwise it will not be degraded. So, the degradation actually happens is at the proteasome. This was discovered by Alfred Goldberg and Martin Reschner in the year 1980. It is a fairly large complex. It is a 26S, a 200 kilodalton complex comprising of the barrel and the end caps. So, the barrel has is the 20S component which is 673 kilodalton proteasome, and it has 32 different polypeptides. These polypeptides, they form a hollow cylinder of which 4 rings are formed. Each ring contains 7 subunits. These rings on the inner side of them have proteolytic activity, much like the trypsin, chymotrypsin, etc. enzymes that are found in the digestive system. So, this can be like a digestion bag for degrading the proteins. And the 19S is the cap unit, as you can see here. So, these are caps which are occurring at the both end of this barrel. These 19S complexes are the ones which recognize the ubiquitinated proteins. They have several AAA ATPases and they have the binding sites or recognizing sites for the ubiquitin chains. So, these 19S particles are also responsible for unfolding of the protein which comes in and also of removing the ubiquitin which was tagging the protein all through its journey. These then also stimulate the proteolytic activity and channel the protein which is incoming towards the 20S particle for further degradation. If these 19S particles are removed, then the ends of the barrel will squeeze and will not allow a larger volume or larger size of protein to pass through it. Also, the proteasome performs a very critical function which is called the processivity. So, this proteasome will not allow movement of fragments larger than 8 to 12 amino acids out of the proteasome. Hence, if it is going to allow release of larger fragments, it may cause aggregation, it may interfere with several signaling process. So, there is a diffusion barrier of 12 to 8 amino acids, and you can see in this diagram that how a protein which is coming out with the ubiquitin tag is recognized and it is fragmented by the 20S into small peptides. So, again this diagram is showing the enzymatic role of the central cylinder and the recognizing and unfolding role of the 19S cap units. The active sites recognize the protein and degrade it into smaller fragments. So, this is summarizing the ubiquitin proteasome pathway, where the ubiquitin which was recycled within the system is now activated by the ATP. Then E1 enzyme bound to the activated ubiquitin will transfer it to the E2. E2 and E3 will now bind to the substrate and attach the ubiquitin. E2 and E3 will undergo several cycles to attach several ubiquitin molecules depending on the target and the destiny of the protein, and this condemned protein will be channeled to the proteasome, where ubiquitin will be removed by the 19S subunit and the protein which is going to be unfolded and channeled into the proteasome for further degradation, and these fragments will be then released. So, ubiquitin is not just a degrading pathway, it is also regulating several key processes in the metabolism. So, the processes such as antigen processing, apoptosis, viral infection, signaling, X inactivation, DNA transcription and DNA damage, modulation of cell surface receptors, ion channels, secretory pathways, then histone ubiquitination in case of heterochromatin formation, and then degradation of regulators which are found in the cell cycle. So, also in the degradation of P53, which is the anti-tumor protein, and in several CDK inhibitors, transcriptional factors and signal transducing molecules. So, all of these processes require ubiquitin processing. With one example, we see that cell cycle depends largely on how the cyclin concentration is increased or decreased at critical checkpoints. So, there are several checkpoints where the cyclin B degradation or cyclin B synthesis is required. At these two checkpoints, cyclin degradation is undertaken by ubiquitin. In immune response, antigen processing by proteasome is also undertaken by ubiquitin. Here the activation of the immune system leads to the release of the cytokine interferon gamma, and this interferon gamma causes 3 of the subunits of the core particles to be replaced by the subunit The peptides generated in this altered proteasome are then picked up by a transporter associated with antigen processing, and then they are transported into the cytosol towards the endoplasmic reticulum, where they enter the groove at the surface of class 1 histocompatibility molecule at the MHC. So, by altering the proteasome, a different kind of peptides are formed, and these peptides will be presented by the MHC molecules, and then they can be recognized by the T cells. So, this is one important function of proteasomes, but by modulating the proteasome structure itself. Many a times use and unuse of a certain organelle can also invite how it is degraded. When we exercise a lot, we make lot of muscles, but if we do not exercise, then there is muscle wasting. Many diseases are also called the myopathies, see that there is muscle wasting. So, here also in remodeling, we encounter the role of proteasome and ubiquitin pathway. So, remodeling will involve proteolysis of the set of structural and metabolic protein and its replacement by other specialized or different proteins for different purposes. Mainly in the bone reabsorption process, there is creation of high concentration of degraded collagen fragments. So, in addition to the calcium and phosphate within the reabsorption lacuna, which are taken up by the osteoclast and then transported to the cell and released. During transcytosis, these collagen fragments are further degraded by cathepsin K and TRAPS. So, these are responsible for osteoclastic remodeling. Various conditions can occur if there is either a mutation in the cathepsin, so which will cause defect in how collagen is degraded, and it will result into the condition called osteopetrosis. Even the viruses, which are known to be very cunning in how they modulate the host machinery, they also fool the system by shutting down or hacking the ubiquitin pathway. So, many proteins are modified by the viruses so that they escape the ubiquitin degradation pathway. Even the human immunodeficiency virus HIV encodes certain proteins VIF and VPU that will interact with distinct Cullen ring like which are E3 ligases to promote ubiquitination of certain cellular proteins, and that will help in spread of infection. Herpes viruses also encode certain ubiquitin ligase that will surpass the cellular ligases, and these viruses will encode ubiquitin ligases which will downregulate those immunoreceptors such as MHC class 1 etc. from the surface of infected cell, and hence evasion can occur. So, now the MHC will not be able to show the fragments of the diseased cell, and it will escape the T lymphocytes, and hence disease will propagate within the host. So, there is a wide scope of research, and this field was opened up by Aaron Scheisenover and Avram Hershko when they discovered ubiquitin in the year 2004. So, as we have seen that several pathogenic bacteria also rely on their ability to tap into the ubiquitin processes, and they modulate the host machinery to spread the infection and not getting recognized by the immune cells. Immune responses can be suppressed through the inhibition of NFK beta signaling system. These transcription factors, they activate genes involved in the immune response, but when they are inhibited by the inhibitor IK beta, they will not show a immune response. So, what happens? The immune response is triggered by ubiquitination of IK beta, but if IK beta escapes ubiquitination, then there will be no large scale immune response. Certain bacteria are able to interfere with this system and then they prevent ubiquitinylation of IK beta. So, there is going to be evasion of IK beta from degradation, and then immune responses will be suppressed by inhibiting the NF-k beta. There are several diseases also which are attributed to the ubiquitin proteasome pathway. So, the ubiquitinated protein accumulation is occurring in various neurodegenerative diseases, in several cancers, and also in hereditary disorders. So, let us look at the role of E3, and E3 ligases are a current favorite in understanding disease regulation and drug therapies. So, E3 ubiquitin ligase, they occur as scaffolds for bringing in the E2 enzyme, facilitating the transfer of ubiquitin from the E2 towards the protein. They are also responsible for having a domain which will recognize the degrons on the protein, and hence they are doing the two functions of recognizing as well as attaching the ubiquitin tag or elongating the chain. So, these E3 ligases have a very crucial motif, which is the ring motif, and this ring is having a zinc in its motif. So, there are several such ligases and they have been attributed to various cancers. So, these Cullen ring ligases such as SCF, CRLs and anaphase promoting complexes have fundamental role in controlling the eukaryotic cell cycle, and if the cell cycle is disrupted, it can result in several cancers. So, these SCF E3 ligases, they have several interacting proteins, namely the S-phase kinases or SKPs, the Cullen proteins and X-box proteins, and they determine if the protein is getting degraded or not, and any mutation or alteration in these factors will result in disruption of the cell cycle. Even overexpression of these SCF complex can be implicated in human cancers. We will also look at other aspects of neurodegenerative and viral, but let us first focus on the cancers. So, you have the CUL4A gene, which is highly increased or amplified during the breast cancer. Then we have the SKP2 oncogene, which is the F-box protein, which is responsible for P27 for degradation. Other such proteins are also F-box protein like CDC4, which targets cyclin E for degradation, and it is found to be responsible in breast cancers as well as endometrial cancers where it is mutated. Also, BRCA1, which is a ubiquitin ligase complex, has been studied regularly and for a long time in cases or in studies or in research for breast cancer and cervical cancer. Also, MDM2, which is also a E3 type of E3 ligase, it is responsible for inactivation of P53 as a result of overexpression of HDM2. So, when there is levels of P53 tumor suppressor protein, and if the levels are going to be regulated or down regulated, then it can cause inactivation, and this MDM2 ligase has been found to be responsible for about 50% of human cancers. Parkin is another ubiquitin E3 ligase which is responsible for Parkinson's disease, and it in the mutated form this will result in defect in the ligase activity. So, these genes and ligases have been implicated in several cancers, and they are now part of strategies to find cures for several of these diseases. Even the neurodegenerative diseases, we had discussed that there is occurrence of plaques and fibrils or tangles which are escaping the degradation pathway. Even in the case of Parkinson's, we have discussed there is a defective ubiquitin ligase, and in this because there is no ubiquitination, there is evasion or escape by the substrate to get degraded, and it gets accumulated and causes a dump into the cell system. Alzheimer's also, where we encounter neurofibrillary tangles and plaques, and here the amyloid beta protein is getting aggregated and ubiquitinated tau protein is also escaping degradation. So, there is key step which either gets missed during ubiquitination will result in these diseases. Huntington disease is also showing one example where a polyglutamine repeat which is occurring in the protein causes it to evade the degradation system. So, no matter you try to degrade this protein aggregate, it will evade degradation. Current research is going that suggests that even the metabolic diseases such as diabetes or insulin resistance which is found in diabetes can also be attributed to ubiquitin. So, ubiquitin has actually opened a Pandora's box towards understanding disease and therapies. So, in case of insulin resistance, there is regulation of the stability of the protein which is the insulin receptor substrate. If this insulin receptor substrate which are adapter molecules between the receptor tyrosine kinase and further signaling molecules, when the IRS protein is knocked out or when it is faulty, it will result in diabetes. And how ubiquitin is responsible here? So, IRS2 is regulated by phosphorylation and by ubiquitin mediated degradation. If it is ubiquitinated, it will get degraded and will not be able to regulate glucose in the blood. So, because IRS2 is responsible and it is sensitive to the blood glucose levels. As we have been discussing, as we have discussed that ubiquitin is a Pandora's box for various therapies and there are potential targets for proteasome and ubiquitin. A drug which has been discovered in the year 2003, and in fact this drug itself this drug itself is a prelude to the ubiquitin Nobel Prize. So, in the year 2003 when the drug Velcade or bortezomib was discovered, it opened up another field of cure for cancer, and it prompted that the Nobel Prize for the year 2004 be given to discovery of ubiquitin. So, it is a long list of various molecules which are now under stages of phase 1 and phase 2 trials and approvals, and they are a harbinger for treatment of various cancers. And we can say that ubiquitin system can give us lot of insight into not just understanding various biochemical metabolic pathways within the cell or the various diseases that we did not know were related to this system, but also open up a new field of understanding drug targeting and therapies which can regulate either the proteasome for better degradation of protein or for creating those signals which can regulate pathways which have become mutated or have become disrupted during diseases. So, we can say that ubiquitin itself being a Nobel Prize material has further lot of scope for research. Thank you.
With this note, we would like to thank Dr. Nati Gulati for giving precious inputs to the lecture, and dear friends, we believe that this lecture appears to be very, very beneficial. So, if you want to give your feedback for this particular lecture, then you can mail us at info.cc@nic.in. And yes, this lecture is going to be uploaded on YouTube very soon for you. So, keep watching the lectures and keep giving your feedback. So Dr. Gulati, what are we going to study in the next session, in the next lecture?
As we have discussed, we are talking about ubiquitin proteasome pathway, which is a fairly selective pathway. We will talk about lysosomal pathway, which is also very selective, and we will see that there is even role for ubiquitin in targeting protein for lysosomal pathway. In the next lecture, we will talk about what separates proteins that are targeted for ubiquitin and for lysosomal, and why there are two pathways that are existing. And lysosomal pathway, as others may feel, is not a very sensitive pathway, but we will see how targeting of proteins towards this pathway occurs, and in what cases lysosomal degradation will occur.
Thanks, Dr. Gulati. Thanks again. And yes, dear friends, keep watching us, keep writing us. We would be meeting again soon. Till then, take care. Goodbye. Thank you. Thank you so much.
Thank you.